Hello students, good day once again, compliment of the season. Welcome to Blood Biochemistry and Immunochemistry BCH 408. Today we are in topic 2, that is metabolism and regulation of heme. Our focus for this study will be to identify the materials needed for heme synthesis, recount the steps in the anabolic pathway of heme synthesis, comprehend the pathway for catabolism of heme, have an insight into control of heme biosynthesis, and finally have some ideas of heme disorders. The question is, what is heme metabolism? Heme metabolism simply refers to the anabolic and catabolism of heme, that is, the synthesis and degradation of heme. We have heard of hemoglobin and myoglobin. What are they and how do they differ? Hemoglobin is a tetrameric protein found in red blood cells where it stores and transports oxygen, while myoglobin is the same but with a difference in that it is a monomeric protein as against the tetrameric for hemoglobin, and it is present in the muscle cells where it also helps to store oxygen. So while hemoglobin is in the blood, myoglobin is in the muscles. Now we talked about pyrrole ring. What you see here is the structure of pyrrole ring. The first item there and the second item, they are the same. I'm only trying to show you the different way they can be represented. How about porphyrin? What is porphyrin? Of course, you know what a pyrrole ring is. I've shown you before. Now look at the structure before you. You see the figures 1, 2, 3, and 4. Those are the pyrrole ring structure. And you see letters A, B, C, and D. Those are the methyl groups being linked with carbon, forming an enclosed structure of porphyrin. So, porphyrin is a four-ring structure, that is, four-membered closed structures containing one nitrogen and four carbon atoms linked to each other by a methyl bridge or methyl bridges. Another thing we have to know or acquaint ourselves with is protoporphyrin 9. These are all important because we'll come across them in our discussion of heme. Protoporphyrin 9 is a cyclic tetrapyrrole that consists of porphyrin bearing four methyl substituents at positions 3, 8, 13, and 17, two vinyl substituents at positions 7 and 12, and two carboxyethyl substituents at positions 2 and 18. Now, this is how the structure looks like in this figure here. Having said all this, all put together, we have something like this. This is a heme molecule containing the methyl, vinyl, and propionyl groups attached to it. Now, before we go into the biosynthesis of heme proper, we have to know certain facts about the synthesis of heme. It is compartmentalized, that is, from one compartment to another. In this case, the synthesis moves from mitochondria to cytoplasm and back to mitochondria for completion of the pathway. Now, the pathway is displayed here. You can see the arrows directing the paths of the ways or reactions. Heme is synthesized in erythroblasts and reticulocytes of the bone marrow that still contains mitochondria and liver cells. Now, let's look at the intermediate structures of heme biosynthesis. From the mitochondria, like I mentioned earlier, the synthesis of heme commences with succinyl-CoA and glycine coming together, reacting together. That reaction is catalyzed by ALA synthase. And what the product is 5-amino levulinic acid. In short, the short form for the word is ALA. So, succinyl-CoA and glycine catalyzed by ALA synthase will give you ALA, 5-amino levulinic acid. That reaction occurs in the mitochondria. Now, that is the rate limiting reaction, after which it migrates out to the cytoplasm, where two molecules of ALA condenses together with the elimination of a water molecule to give you porphyrin. That reaction is catalyzed by ALA dehydratase. Now, 4 molecules of porphyrin, that's PBG, will condense with the aid of PPG deaminase to give pro-uroporphyrin molecule. That is, with the removal of 4 molecules of amine group. Pro-uroporphyrin will again react to give us uroporphyrin 3. The reaction is catalyzed by uroporphyrin 3 synthase. Uroporphyrin 3 will undergo decarboxylation reaction in the presence of decarboxylase to give us co-proporphyrin 3. Co-proporphyrin 3 will undergo isomerization reaction to give us protoporphyrin 9. protoporphyrin 9 will be acted upon by an oxidase, that is protoporphyrin oxidase, to give us protoporphyrin 9. Remember, there's a distinction between protoporphyrin 9 and protoporphyrin 9, as you can see. The last reaction here is protoporphyrin 9 being acted upon by ferrochelatase. That is the enzyme that catalyzes the addition of ferrous ion into heme molecule, and the result is heme. Now let's look at the summary of heme biosynthesis. There are 4 pyrrole rings with metal in the center of heme. That metal is mainly ferrous ion in hemoglobin and myoglobin. Heme is synthesized partially in the mitochondria and also partially in the cytoplasm. There are 8 steps in total in the biosynthesis of heme. These 8 steps are equally divided at the 2 locations. There is a need for proper control of heme biosynthesis to maintain cellular hemostasis. So it is important that the biosynthesis of heme is regulated. As part of the regulation of heme biosynthesis, let's look at the subcellular localization of heme regulation. Now, the enzyme synthase is synthesized in the cytoplasm and transported into the mitochondria, where the substrate succinyl-CoA is produced. Succinyl-CoA is produced within the mitochondria, but the enzyme that is required to catalyze that reaction is synthesized in the cytoplasm, which means that enzyme has to be transported into the cytoplasm. So, if there is no mobilization or transport of the enzyme into the mitochondrial compartment, the synthesis cannot occur. In the erythropoietic cell, heme synthesis is coordinated with globin synthesis. If heme is available, globin synthesis proceeds. If heme synthesis is absent, AMP-independent protein kinase is active. The kinase phosphorylates and thereby inactivates the eukaryotic initiation factor 2. This prevents further globin synthesis. Certain drugs and steroids can increase heme synthesis through increased production of the rate-limiting enzyme synthase. One of the key regulatory steps is the feedback inhibition of synthase, as shown in the diagram here. When heme levels are sufficient, it binds to synthase, inhibiting the activity and preventing the overproduction of heme molecule. Let's look at the relevance of heme. It is an essential prosthetic group in some proteins necessary to perform diverse biological functions. Examples, as mentioned earlier, are hemoglobin and myoglobin. Some enzymes use heme as prosthetic group, such as co-cytochromes, catalase, and nitric oxide synthase. Some functions of heme include 1. As reservoir of iron. 2. As electron shuttle in electron transport system or chain. 3. They function in cellular respiration by transporting oxygen. 4. Cellular differentiation and proliferation. They also act as cofactors, transport of oxygen in hemoglobin, storage in myoglobin, and prosthetic group for cytochrome P450 enzymes. There are different forms of heme. We can talk of heme A that exists in cytochrome A and heme C that exists in cytochrome C. Both heme A and C are both involved in oxidative phosphorylation process. Now we come to the disorders of hemosynthesis called porphyrinias or porphyrias. Now these are occasioned by deposition of porphyrin pigment in tissues. This is caused by deficiency in one of the enzymes involved in the biosynthetic pathway, or absence of porphyrin precursors, or accumulations of porphyrin metabolites. Most forms of porphyrinuria or porphyria are caused by a gene change or mutation passed down from one or both parents. Porphyria or porphyrinuria can occur if inherited as autosomal dominant pattern, autosomal recessive, or acquired form. That is, it may be triggered by certain other factor. In autosomal dominant inheritance, one copy of the gene in each cell is mutated. This single mutation is sufficient to reduce the activity of an enzyme needed for heme production, and this increases the risk of developing signs and symptoms of porphyria. In autosomal recessive inheritance, both copies of the gene in each cell have mutations. Most often, the parents of the individual with an autosomal recessive condition each carry one copy of the mutated gene, but do not show signs and symptoms of the condition. In acquired form, certain factors can trigger the buildup of porphyrins in tissues, such as too much iron in the body, viral infections such as HIV and hepatitis C, heavy alcohol use, smoking, and even excessive hormones. Now, types and enzyme deficiency in porphyria. The figure shown here gives the condition, the enzyme deficiency, and the type of inheritance. Now, this is the pathway for the heme catabolism or heme breakdown. The enzyme required here is heme oxygenase. Heme oxygenase to give us biliverdin. Now, we also require biliverdin reductase to give us bilirubin. Now, the bilirubin is acted upon by 2-UDP glucuronic acid in two-step reaction to give us bilirubin diglucuronide. Heme degradation begins inside macrophages of the spleen, which remove old and damaged erythrocytes from circulation. Red blood cells are engulfed by cells of the reticuloendothelial system. The globin is recycled into amino acid, which in turn are catabolized into intermediates of the citric acid cycle and fatty acid oxidation. Heme is oxidized. The heme ring is opened by heme oxygenase. The oxidation occurs on a specific carbon, producing the linear tetrapyrobiliverdin, ferric ion, and carbon monoxide. In the next reaction, second bridging methylene is reduced by biliverdin reductase, producing bilirubin. The green pigment is thus converted to the red-orange bilirubin. Bilirubin is then transported in the serum by albumin to the liver, where it is conjugated with glucuronides by biliverdin glucuronide transferase and excreted in the bile. In the intestine, bilirubin is deconjugated and converted to urobilinogen and stecobilin. Some urobilinogen is reabsorbed and excreted as urobilin in the urine. Most urobilinogen is oxidized in the feces to stecobilin, which gives feces its color. Now, this is a summary of the discussion. You can see from the picture here the action of the RBC, red blood cells, the action of macrophages, how heme is degraded to amino acid and to heme, to porphyrin, then conjugated, unconjugated, and then as some are even stored and others released to transferrin. Now let's look at the significance of heme degradation. Free heme concentration greater than 1 can be toxic. Greater than 1 millimolar can be toxic because it catalyzes the production of reactive oxygen species. To cope with this problem, heme degradation is very crucial for the body. In animals, this pathway is an excretory system by which the heme from the hemoglobin of aging red blood cells and other hemoproteins are removed from the body. Products of degradation such as carbon monoxide act as a cellular messenger and functions in vasodilation. Other metabolites of heme also have additional important functions and are involved in various critical cellular events. Heme degradation is believed to be an evolutionarily conserved response to oxidative stress. In higher plants, heme is broken down to phycobilin protein, phytochrome, which is involved in combating light responses. In algae, it is metabolized to the light harvesting pigments, phycocyanin and phycoerythrin. Phycocyanin and phycoerythrin. Thank you for your time and for listening. Once again, compliments of the season. As regards this lecture, if you have any concern, please, you can post. Do the normal posting through your class coordinator. He knows how to get it across to me. And very soon, our physical class will take place. Please, before then, read and study hard because the tests will come very soon. Thank you.